All right, give me 1 second. Hopefully you're seeing my, oh no, I need to share my screen. All right, so hopefully you're looking at my screen now. The last time we took a look at these notes, we had finished talking about the replication process. So the main enzymes that we had discussed were primarily helicase, which we see right. Let me just go ahead and put the slideshow on. All right. So the last time we talked, ladies and gentlemen, we had looked at how replication actually occurs. Somebody is in the chat, for the love of. Okay. And then one of the things that we had discussed was how DNA polymerase does the job of placing the new nucleotides into the brand new DNA. So we again over here on the side, on the right hand side, we see the original DNA. We see this unzipper, or this zipper sort of enzyme right here that is helicase. The helicase goes in and unzips the DNA into the two sides by breaking the hydrogen bonds, creating the replication fork. And then, of course, we have the DNA polymerase, and we can see it moving from the 5 to 3 direction on the top, making a full strand of DNA, and it is matching up the complementary nucleotides and also doing the proofreading. So again, DNA polymerase is like the builder. It is going to build my new DNA strand. And again, it does this by adding new nucleotides to the DNA. Remember, we are replicating our DNA before cell division because DNA has to be in every living cell. It's one of those like characters of life things that we've talked about all year. And please also do remember there are 3 main functions of DNA. The DNA has to be able to store genetic information, and it has to be able to copy genetic information. So that's what we're talking about here. We're copying the genetic information. The rate of elongation in a nucleotide is 50 nucleotides per second. That's really fast. But the cool thing is, at the time that the DNA polymerase is actually reading and placing the new nucleotides, it's also proofreading it. And we know that DNA replication is really, really fast. And we know that it is really, really accurate. It is so accurate, in fact, that the DNA polymerase makes about 1 mistake for every 10 billion nucleotides it places. That's like really, really accurate. All right, so we're going to go ahead and continue. So this is sort of where we left off last time. So this is sort of the new slide now, which in your page of notes, I want to say, let me pull up mine so I can see it really quick. This should be on the 4th page where it says enzymes proofread DNA. So that's where we're going. Enzymes proofread DNA. What does that mean? It means that as we are adding nucleotides to the growing DNA, there is mistakes that are made. Okay, about 1 in every 10,000 base pairs. The problem is, ladies and gentlemen, that would end up leading to a lot of mutations. So, as it is adding them and making those mistakes, at the same time, the DNA polymerase is proofreading as it's adding nucleotides. So just like when you proofread an essay or when you proofread like a paragraph submission, that when you're doing a short answer question or something like that, you read through it to make sure that it makes sense. The DNA polymerase is doing the same thing. It's saying, okay, did I follow the rules for base pairing? Are my A's all attached to T's? Are my T's attached to A's? Are my C's with the G's? Are the G's with the C's? If it makes a find a mistake, it's going to remove the mistake and then continue on. Now, there is actually another enzyme involved called an exon nuclease. An exon nuclease, and I always remember this because the word exon has the word X in it, and to me, the letter X always looks like a pair of scissors. The exon nuclease actually is the one that does the cutting. So it cuts out any mistakes, and it will then, the DNA polymerase then comes along and makes the correction and continues copying the DNA. So it's kind of like if you're typing and you make a mistake and you just hit the delete key, or you go back and hit the cut and you cut something out key. That is sort of what the proofreading is like. When it finds a mistake, it just deletes it, fixes it, moves on, just like you do whenever you're working with a computer. After we continue on with our proofreading, we go from 1 in 100,000 mistakes to 1 in 10 billion nucleotides in terms of a mistake, because so many errors are actually corrected before the replication is completed. Okay. In terms of percentages, this is like if you went through the test the first time and got a 1%, and then you went back through and made corrections and you came up to a 99.9% just because you made corrections. Okay. That is the rate at which we're making corrections. We are making a lot of corrections as we go along. All right. So there can be accidental changes. I mean, there's still this 1 in 10 billion nucleotides that's going to be matched wrong. So maybe an A gets matched with an A, or maybe an A gets matched with a C, or maybe a C gets matched with a T, right? Whenever we say that there is a mistake in the base pairing, that is what is known as a mutation. Now, in movies, mutations, these mistakes in DNA, um, always usually end up with somebody having superpowers, right? So like, oh look, I have a mutation. I can now turn invisible, and in the X-Men, oh, I have a mutation, and I can now like read people's minds. In reality, when you have a mutation, normally this leads to, in many cases, nothing. Like it's just an inactive gene, so it's not a big deal. But in other cases, you might end up with a change in the DNA code, which means there is going to be a change in your traits. There is going to be a change in how those traits are expressed. There is going to be a change in which proteins are created. That, again, is a mutation. So, if the DNA how does DNA become mutated? First of all, it can be damaged. We have the ability to damage our ability to replicate DNA. We have a damage to DNA when we are exposed to chemicals, radioactivity, X rays, ultraviolet light, and certain molecules in cigarette and other smoke. And I say and other smoke because we know this because firefighters who obviously deal with a lot of smoke for a living um, end up having higher rates of lung cancer than the general population. So it's not just cigarette smoke, it is pretty much all smoke that you should not be ingesting. Um, but as these changes are occurring, your cells are monitoring themselves and they are trying to repair the genetic material. Remember that DNA polymerase, its job is to proofread. Um, and generally, we find so far, and again, this is so far because science is always evolving, there are about 130 DNA repair enzymes that we have identified. There are literally enzymes that exist in your body whose sole purpose is to go in and fix damage to your DNA. Okay? That's a good thing. All right, so how do we go about repairing the damage? Oops, I'm going a little fast. First of all, we detect the damaged section of DNA. So obviously we can see it here. I have this kind of blob thing that's sort of there. Maybe that is a mutation created by a mistake. Maybe that is a mutation created by exposure to radiation. Maybe it is a mutation created by whatever damaged my DNA. Chemicals, right? So as the repair enzyme is going through and checking for damage, it identifies what section of my DNA has been damaged. We then send out the damaged section is end up is going to be cut out. And again, this is the exon nuclease. I'm not sure why I did not include that in there, but that is the exonuclease. Exo, of course, is a prefix that means out. So basically, it's the nuclease that cuts things out. Exonuclease. It's right there in the name. So the exonuclease comes along and cuts out the damaged section. Ding, ding, ding. That's a good thing. Um, and then the DNA polymerase, our buddy, the builder, comes back in and rebuilds that section properly. And then we have ligase, which is kind of like a glue. Ligase literally will just kind of seal everything back up together. So it's going to seal the free ends of the new DNA to the old DNA, and it makes the strand complete. So that pretty much covers it. So this repairing of damage can happen when there is a mutation, any mutation. And again, all a mutation is a mistake or damage to your DNA. Now, these mutations can happen naturally because the DNA polymerase made a mistake, which doesn't happen too often. More often than not, we get mutations from environmental factors, which can include anything from radiation to chemicals to whatever, okay, to smoke, anything that can damage your DNA. Either way, we do have these sort of safeguards in place to help restore and repair the DNA. Because again, if you have entirely too many mutations happening that are not going to be repaired, it can and will, in many cases, lead to serious illness in a living organism and to up to and including the death of that organism. And of course, the biological imperative of every living thing is to stay alive. All right, so that covers or finishes up, I should say, these notes. Let me get out of the video. Give me one moment.